Experience tranquility as you drive through the population-free lush green slopes of Vagnakot Solan to reach functionality and aesthetically spread 25 acres of easy-flowing and community-friendly landscape. Welcome to JP University of Information Technology, Vagnakot Solan. Set up in 2002 under the extraordinary Gazette notification of Government of Himachal Pradesh. This is the only private university in the state whose chancellor is the Governor of Himachal Pradesh. With 108 professors, most of whom are PhD from reputed universities around the world, JUIT offers BTech, MTech, MSc, PhD in computer science, IT electronics and communication engineering, bioinformatics, biotechnology, and civil engineering, whose curriculum is supplemented by SWAYAM and PTEL. A home to almost 2,000 students, this university is twice accredited by NAAC. The world-class state-of-the-art infrastructure of JUIT has 57 labs, including Virtual Lab, eYentra Lab, IoT, lecture theaters, classrooms, tutorial rooms, laboratories, vast auditorium, administrative and faculty offices, an enormous library, and 6 well-equipped gyms. The campus is designed with a vision of creating a university of international repute and highest global standards. Fully equipped with learning tools and web-based educational environment, the campus is planned for the advanced computing infrastructure. Facilities like bank, temple, and helipad are all within the campus. JUIT takes pride in being a member to IEEE Chapter and Computer Society of India Chapter. It also conducts student exchange programs with the University of Nebraska, Omaha, and University of Florida, USA. To give an experience of the real corporate world and understand the challenges that lies ahead, our students also get an opportunity to connect with industry professionals and self-made entrepreneur. Our exclusive placement cell has an excellent placement record. All this has brought us several accolades. We're ranked among the top 100 engineering institutes by NIRF, MHRD. We've been declared Best Private University of Himachal Pradesh by Dialogue India in 2018 for academics and infrastructure. We're also ranked first under category of value for money and third under the category of top private university in 2018 for offering value for money education by India Today. With a hygienic mess named Annapurna, well-furnished hostels with single and double rooms, mechanized laundry, 24/7 water supply, complete power backup, and Wi-Fi connectivity, we ensure that students feel at home while they're away from home. The overall growth in personality and confidence is taken care of by conduct of debates, declamation contests, and a host of indoor and outdoor games. NCC Wing for Girls, and a vibrant youth club that organizes popular sports festival called Padakram, literally festival and cultural festival, which further heightens their energy levels. They're also encouraged to develop independence in thought and action, as well as trained to deploy appropriate technology paradigms for given tasks, explore new horizons, and lead teams to solve complex problems. With our learner-centric educational process, students are free to find their own paths and learn their way to limitless opportunities that awaits them. So be a part of JP University of Information Technology, Vagnakot, Himachal Pradesh, to be the leaders in innovation, entrepreneurship, creativity, and management. Because this is where we engineer lifelong learners.
Good afternoon to one and all present here. I, Dr. Tiratha Raj Singh, on behalf of Center of Excellence in Healthcare Technologies and Informatics, Department of Biotechnology and Bioinformatics, JP University of Information Technology, Vagnakot, Solan, Himachal Pradesh, India, welcome you all onto the second season of our international webinar series. And this is our third webinar of this particular season. Last year, we have organized 6 webinars, one every month, and that was the compilation of our season 1. Now, this is the third webinar in the season 2. And you know, the Center of Excellence was established to do some research and academic related activities in the domains of biomedical informatics, bioinformatics, and healthcare sectors. Before moving further, first I would like to invite our head of the department, Professor Sudhir Kumar, to give you a brief about our department and its activities, and then we will move further. Professor Sayal, please.
Thank you, Dr. Tiratha Raj. Am I audible?
Yes, sir.
Okay. So, welcome to all present here in this second international webinar series of our Center of Excellence in Healthcare Technologies and Informatics. And as Dr. Tiratha Raj conveyed you that I can have a brief on the department activities and about the department. So this Department of Biotechnology and Bioinformatics is associated with the JP University of Information Technology since we are here 2002. So since the inception of the university, the department exists, and the department, as the name indicates, having two domains, broad domains, like one is biotechnology, another is bioinformatics. And in these domains, we offer the students BTech degree in biotechnology and bioinformatics, and we also offer masters in biotechnology, microbiology, and MTech in biotechnology, and PhD in bioinformatics and biotechnology. Our department is having very strong alumni base, and due to that, you must have seen in the international webinar series 1 and even in 2, many of our alumni, they are sharing their experiences and their thoughts and their current profiles and their jobs, their knowledge. So this is a very good platform for sharing knowledge and getting ahead in life of academics and research. So I'm very sure those who are here and they have been to our earlier seminars too. So these seminars are very useful, and they give us a glimpse of the activities of the modern tools and techniques which are going on, especially in the area of bioinformatics and healthcare technologies and medical informatics. So I also welcome today's speaker of the day, and hopefully you people will enjoy the seminar. And I again welcome all of you, and for your time. All the participants, I'm very sure that this seminar for you will be very useful. And if you would like to have any further information about our international webinar series in future or in past, you can browse the center's web page also. And also for other activities, you can have our Department of Biotechnology and Bioinformatics web page. And you can see here a beautifully designed logo also for the Center of Excellence in Healthcare Technology and Informatics. So this logo is also designed through a competition by our BTech students. So we have a very active student body of graduate or undergraduate students. So I'm very sure almost we are ending the COVID pandemic. So once it's over, because it is still around, but hopefully we are through this within maybe few months. You are most welcome. Please visit our campus, and it is one of the best beautiful 12 academic campuses in the India, and you can see and browse it. And I'm very sure when you'll come here, you will see the nature beauty, and we are almost midway between Solan and Shimla. With these words, I would like to see you physically present here with us anytime in future. Please now enjoy the seminar. Over to Dr. Tiratha Raj.
Thank you very much, sir, for the fruitful information for all of us. Now, I would like to request Nissan just to play a small video to reflect the activities of CEHTI. This was the girl that designed the logo. Okay, thank you, Nissan. Now, I would like to call upon one of our students. She is going to introduce today's speaker, Dr. Rajendra Gupta. So, first, I would like to call upon Tiasa to introduce Rajinder.
Hello, sir. Am I audible?
Yes.
Rabindranath Tagore once said, "The highest education is that which does not merely give us information but makes our life in harmony with all existence." A very warm good afternoon to one and all present here. I'm Tyasha De, currently pursuing my second year of Masters in Biotechnology from the JP University of Information Technology, Solan. I'm enthused to welcome each one of you to the International Webinar Series, Season 2, Session 3, organized by CEHTI. I would like to take this opportunity to introduce our guest lecturer, Dr. Rajendra Gupta, currently affiliated with the ME Noethel Group for Computational Microbiome Research, Technical University of Munich, Germany. Dr. Gupta received his bachelor degree in biotechnology in 2007 at DYP BBI University, Pune, India. For his bachelor's thesis, he worked on the toxicity effects of novel plant extracts. He then joined JUIT Solan to pursue his master's degree in computational biology, where he explored network motifs in cancer pathways. In 2014, he joined the National Agri-Food Biotechnology Institute, Mohali, as a junior research fellow and worked on the development of Universal Biomolecular Relationship Database, extracting and collecting biological interaction data from databases and scientific publications. In May 2016, Dr. Gupta joined the Department of Toxicogenomics at the Maastricht University as a PhD scholar. He worked with RNA sequence data for human cell models through established approaches and explored new avenues. He is currently working as a postdoctorate in the Scrimmer lab at the Technical University of Munich. As a part of his postdoctoral work, He's investigating and developing tools and pipelines for microbial meta-transcriptomics data. Today, Dr. Gupta will enlighten us on FUSE, a tool for functional analysis of RNA sequence data. A very warm and hearty welcome to you, sir. I'd also like to take a minute to welcome the head of the department, Professor Dr. Sudhir Kumar, the webinar convener, Dr. Tiratha Raj Singh, and the webinar coordinators, Dr. Nishant Jain and Dr. Rna Raj Singh. Now, I request Dr. Gupta, sir, to start the talk and enlighten us. Thank you.
Thank you, Tia sir. Now, platform is all yours, Rajinder.
Okay. So first of all, thank you very much for the invitation, sir, Tiratha sir, and Tiasha, thank you very much for this wonderful introduction. Now I will start sharing my screen. Give me a second. You can see my screen now?
Yes.
Yes. Okay. So good morning, everyone. Good afternoon, good evening, based on your time locations. Thank you very much. Thank you again for this invitation. So today, as Tiasha already mentioned about it, so I will talk about FUSE, a tool for the functional analysis of the RNA-seq data. So currently I'm a postdoc at the Technical University of Munich, but today I will show you my work from my PhD. So this was a tool which I developed for advancing the analysis of RNA-seq data. So I will go through each step of this tool, like what were the problems that I faced working with the RNA-seq data, and what was the motivation behind development of this tool, and what are the different steps to for this tool to run, and then maybe after that you can use this in your work as well. And then if you have any questions in between afterwards, you're most welcome. Okay, so let's dive into it now. Okay, so outline of my presentation. So I will start with what is RNA-seq data. Then I will talk about gene and the transcript expression, what are the problems, and then what are the steps of development of this tool called FUSE, and then a final assessment of the tool, that just to show that this works or not work, right? There are a lot of tools which we developed or which we hypothesize, but they don't give us the type of results we want. So that's why we have to do an assessment of the tool towards the end. Okay, so starting with the very basic thing, that's the central dogma of biology. So I think everyone present here knows about it. So, but just to set the base for this slides that will come after this. So we have our DNA. Just let me put my pointer. Yeah. So we know, like in our cells, we have DNA, and through replication, we make different copies of the DNA, and through transcription, the DNA is made into RNA, and then through translation, RNA makes the proteins, right? So this is the basic biology that we understand. And here in this talk, I will talk about RNA, the quantification of RNA, the identification of RNA in our cells, in our tissues. And that's the part where it's called RNA sequencing. So what exactly is RNA sequencing, right? So most of you must know about it, but just again to set a bit of more base to that. So RNA sequencing is a next generation sequencing which helps to tell the presence and the quantity of RNA in the sample in our system, right? And then it tells us like what genes are encoded, like what genes that are encoded in our DNA are turned on or off, right? So this tells us like what genes are expressed in our system through RNA, because when a gene is expressed, that means it is making an RNA. So through RNA sequencing, we can get to find that out. So what genes are expressed in our system. And then when we use that is in conditions where we have to compare normal and diseased conditions, right? So maybe we have a tissue sample from a healthy patient and a tissue sample from an unhealthy patient, a diseased patient, let's say for instance a liver condition. then we want to compare what changes in these two conditions, right? Like we can tell a diseased liver looking at different tests and different pictures of the liver, but like what changes at the gene level. So we want to see that what changes at the protein level, at the transcript level. And then at that time, we can use RNA sequencing to see what genes are expressed more or less in the disease condition compared to the normal. Similarly, we can look for effects of different drugs, effects of different doses, like therapeutic dose, like a toxic dose. So we can see at what doses how the system reacts to it. We can also look into different types of treatments, different types of methods used, any type of comparison we want to make. So we can do it through RNA sequencing and see what changes are happening in the system. Okay, so but what is exactly gene or the transcript expression, right? Like let's see how RNA sequencing works. This is a dummy example which I'm giving you just to have a bit of more understanding of the concept. So let's imagine we have a gene, right? And then we know in eukaryotes, like we human beings, we have alternative splicing, and through alternative splicing, we can make different transcripts from a gene, right? And then let's assume these blue colored bars, these represent coding sequences, and these light blue ones are the untranslated regions. So what happens in RNA sequencing is when we sequence our sample, so we get some reads, right? So these small lines represent reads. So we get some reads, and then we try to, when we do a reference-based genome mapping, so what we do is we have a genome of a species. Let's, for instance, say for a human being that we are trying to see. So then the reads from our sample that we have got from the RNA sequencing, so we try to map it to the known transcripts, right? We try to see where it maps. But in this case, what you can see is that this transcript, this and this one, they all have the same regions. So what reads map to this transcript can also come to the transcript 2 and the transcript 3. So what we do now is we try to find transcripts which are unique. Like in this region, these 3 transcripts are unique for these 2 transcripts, right? This one cannot have it. So when we have 5 transcripts for this region, so these 5 can come from transcript 1, 2, or 3. But then we find out like this region, right? This region has 3 transcripts. So that means out of these 5, 3 come from here, right? Like from 2 or 3, and then the 2 might be coming from here. So let's see. Then we try to find more unique reads. Like in this case, we found 2 reads that map here, 2 reads that map here, and then 3 reads that map here. So after getting these unique hits for these transcripts, we are able to tell how much expression is of a particular transcript. Right now we see for different regions of this transcript, we have these reads, and then they sum together to 10 reads. And this one has no unique reads mapping to this transcript, so 0 reads. And then this one has 21 reads. And then when we put all these numbers together, so this becomes 31, and this represents our gene expression. So the gene expression is a sum total of the expression of all the transcripts. And then the transcript expression is the individual value for each of this transcript. But why does it matter, right? Like why am I talking so much about gene and the transcript expression? So let's see an example. So let's say we have 2 conditions. So say this is the healthy one, this is the diseased one, and then again these are 2 genes, the same genes in 2 different conditions, and then these are the same transcripts. So what we see is that these transcripts have different expression values, right? You can see these values different over the 2 conditions. But then you see that the gene expression is exactly the same, right? So when we put all of these numbers together, that becomes the gene expression. So these numbers are exactly the same. So when we say a gene is upregulated, a gene is downregulated, that means that a gene is expressed more in a particular condition, or gene is less expressed in a particular condition. But then in this case, this gene is not changing, right? Like it has exactly same amount of expression in both the conditions. But then if we start looking at each transcript, we can really see there are differences, right? Like here in this case, this longest transcript was the most expressed one. In this case, there is a shorter one which is more expressed. So this might not be functionally comparable to the longer one, right? So here we can see it has this region missing. So maybe the functional capacity of this transcript is less or maybe different than this bigger transcript. So that's why it's important to start looking at the transcript expression rather than just looking at the gene expression, right? Yeah, so why so serious about it? So yeah. But then also the thing is like these different transcripts, right? So these can be different types of transcripts. So this can be protein coding, this can be long non-coding, this can be pseudogene, it can be T cell receptor. So these can be all different types of transcripts. So when we are putting the sums together, we are just assuming that they all make the same protein, and that's not the case. Even if these are two protein coding transcripts, they have a difference in their sequence, so they won't make the same protein. And this can be the case that the first one is a non-coding transcript. So we also have to take that into account. So we cannot just rely on these summation numbers, because that just hides away a lot of differences that are happening at the transcript level. Then the question is like, then why are we inclined towards gene expression, right? Like if it's so bad, if it's it masks away so much information, so why shouldn't we talk about the transcript expression? Then why there are so many papers and so many studies being done on the gene expression? So the reason being is like the most biological databases that are available, they have the data stored with respect to genes and proteins. So the transcript is still evolving. The knowledge, our knowledge about different transcripts is still evolving. So that's a reason that we don't see a lot of databases that allow us to make comparisons based on the transcript identifiers. But there are some ways to do it, but then there's always a loss of information. So that's why we still have a lot of studies that only talk about the gene expression and very little on the transcript expression. But then does it mean that if we move all these databases to handle our transcript data, resolve all our problems, right? So is that the thing that we should be worried about, right? So I will show you now an example where I tell what are the problems if we only talk about the transcript expression, right? Like so far I told you like what are the limitations with the gene expression. So now let's see if there are any problems with transcript expression. So if we do a study that is based on only transcripts, so this is an example of a TP53, which is one of the most studied cancer gene, which is also known as P53. So here you can see this particular gene, right? It has 25 protein coding transcripts, 3 non-coding, and 1 nonsense mediated decay. Like these are all the transcripts which are made from this one gene, right? Like through alternative splicing. And then a closer look tells that these protein coding transcripts may make same or different proteins, right? Like if you can see, like this is a protein coding transcript, this is another protein coding transcript, and they don't share the same region. So these might be making different types of proteins or different proteins with some different functions or limited functions. And then these two might be making the same protein because they have a lot of overlap on the coding sequences. Maybe the UTRs are different, right? So this is yet to be seen, right? Like for a lot of different genes. So a closer inspection of all these protein coding transcripts tells us like out of these 25 protein coding transcripts that are listed here, so 13 of them make the same protein, right? Like now you can imagine if I start analyzing each of these transcripts individually, I might miss out on the information which this group carries, right? So maybe in certain cases, 2 transcripts, like maybe for instance, these 2 transcripts, which has exactly the same coding sequences in between, maybe the UTR is a bit different. Here you can see that maybe they will make the same protein, but the only difference is in the UTR regions, which might be used by the cell or by the biological system for locating these transcripts or locating these mRNAs at a particular site in the cell, right? But that is not of importance to us. What we want to know is how much protein will be formed, right? And then maybe this is coming from this transcript 220 or 228, doesn't matter. We just want to know how much protein will be there, because protein is finally going to be the functional unit of the cell, right? Structural and the functional unit of the cell, the traditional definition of the protein. Yeah, okay. But then like in this case, we could see that because this is a well-studied gene, right? A lot of knowledge is available for this particular gene. But you can imagine there are a lot of other genes for which we do not know so much. And then when we do not understand the gene, that directly implies that we also do not understand what different transcripts it make and what type of proteins that it will make. And then we also saw that a same or similar proteins can also be coded by different genes, right? So if you see an example of histones or ubiquitin conjugating enzymes, you can see there are different genes, but they're capable of making very similar proteins, which can have the same type of functions. It's just a thing that they are expressed more in different tissues, but they do the more or less the same type of function. But then we have two scenarios now, right? So the first one is that we have a gene expression where we put all the transcripts from a gene, we put them all together. And then we have a transcript expression in which we take all the transcript individually. And they both have their limitations, right? So, but then what should we do about it? Like we cannot go forward with gene expression. We cannot go forward with transcript expression. So this is where I felt like there is a need to make something that takes care of these issues. And then what I hypothesized is, yeah, so let's, okay, so before I have to go to the hypothesis, so let's see visually like what I want to achieve, right? Like what I want to achieve. So let's say, for instance, we have two genes, gene A and B from the same system. They have different protein coding and non-coding transcripts. Let's say the green ones are the protein coding transcripts. Together they make gene expression, individually they make transcript expression. And then we want to extract the protein coding transcripts from these genes, which will make the same protein, right? So we get these ones which make the similar function transcripts. So these ones will have the same function, which means they will make the same type of proteins. So this is what we want to achieve. And now you see like this one comes from gene B, these two comes from gene A. So this is quite possible that different genes are making the transcripts which make the same protein. Okay, so yeah, so what was the hypothesis? So the hypothesis was if we can group our transcripts based on the function, right? And then we will have better biological interpretations of the RNA-seq data. At this point, what we do is we do a locus-based grouping, right? So when we have transcripts coming from the same gene, so that means the same locus on the chromosome. So then we group that. So that is a locus-based grouping. And then here, what I want to do is a function-based grouping. And how can we do that, right? Like how to deal with the functional grouping, what are our options to do that? So the first one is if we know the protein function for all the proteins known for the human beings, because I worked with the human data. So let's talk about the Homo sapiens here. So if we get to know the function of all the proteins, maybe then we can make a global comparison. But that's not possible at this point. Maybe with the alpha coming that the Google is developing, maybe it gets possible in coming years, and it is doable on local machines as well, local servers that we have. But at this point, it's a bit of a dream to have. So protein function availability for all the proteins is not there. And then comes to the 3D structures that we all know. The protein 3D structures is a hard problem to solve. And also we don't have the 3D structure for all the proteins. If we had the 3D structures, maybe we can do a better comparison of these protein structures, but that's also not possible at this point. But then comes the secondary structures, right? Like the sections of the proteins which have these secondary structures which come together to make the whole protein. So we have information on secondary structures. We have a lot of different tools which can make these predictions from the protein sequences, and they are pretty good. They have good sensitivity and specificity. So we have pretty nice tools to do that. And then at the same time, we have a lot of information on the amino acid sequence of proteins. So then taking these two features, so I wanted to develop a way where we can compare what proteins will have the same function. But then, the proteins have an army and we need a Hulk. And let's see if I was able to make one. So yeah, so this is the workflow for Fuse. So I will go into each step in a not in too much detail. So I will give you an overview here, but if you have any questions, feel free to ask at any time. Okay, so we take all the protein sequences for the humans at this point, and then we do a local blast, one-to-one comparison. So all sequences are compared with all sequences. So we have a sequence alignment results for it. And then on the other hand, we use a tool called Interpro, which is an ensemble of 14 different secondary structure and family prediction tools for proteins. So we make domain predictions. What are the structural and the functional domains in these proteins? So we make that. And then on top of it, I developed a module which makes alignment, like similar to the sequence alignment of the proteins. I made a module to make a domain alignment of the predicted proteins. So then it tells us, like in two different proteins, how many domains are in a particular set of order, or number of domains that are present. If the domain numbers are changing, like the count of domains is changing or their order is changing. So it takes into account all of these information and gives us a domain alignment. And then we create from this information on the sequence alignment and the domain alignment, for each protein pair, we create a reference. And from this reference information from the sequence and the domain alignment, we calculate a score for each protein pair, that protein A and protein B, how similar they are. And then we have different scoring schemes. So the discovery score is a lenient one, and the knowledge score is a more stringent one. So this is more towards like when you have to, when you are studying very novel proteins and you want to get more information about them. And then the knowledge score is more stringent, and it helps you when you want to learn more about very specific functions of the proteins. And then we calculate a score for these protein pairs. And then based on the scores, we create our groups. So a very high score. So the score range is 0 to 100, 100 being the best pair, that the two proteins have the exactly same functions. And then we say like all the protein pairs which are overconfidence score of 90. I'm sorry. Okay. So yeah, so we say that all the protein pairs which are over a score of 90, so they will be grouped together. And then we have our functional based grouping, right, instead of the locus based grouping. So when we have a locus based grouping of the transcript, we say that as a gene expression, and then we do a locus based grouping, and then we get our expression data from the experiment.
Excuse me, Jasprit, please mute yourself. Jasprit Kaur.
Okay, thank you. Yeah, so then we calculate our the group expression SFPG, so similar function protein group expression. So this is how we calculate the expression, and then we compare like this expression with the gene expression and see if we get a functionally relevant result using this workflow. So this is an overview of the whole workflow. And okay, so the basic step is initially the data preparation. So this is more interesting if you want to use this tool. So I will very quickly go over this. So what you need to start off with this workflow is to have an ensemble data. So to get all the amino acid sequences for the protein coding transcripts from the ensemble. And then you need to have a local blast installation so that you can use it to make a all versus all comparison of these amino acid sequences. and then you need to have a Interpro installation with you, through which you can do a structural and functional domain identification in your amino acid sequences. And then all other scripts are provided with this tool. They're available on GitHub, that you can generate all the subsequent analysis. Okay, so there were 2 scores which I mentioned, right? So the 2 scores after doing the sequence alignment, domain alignment. So there are 2 scores to calculate how much similar 2 proteins are. I'm sorry. Yeah, so there are 2 scores. So the first one is the discovery score. So the discovery score is a bit of lenient, so it only considers the sequence alignment data. So it doesn't go towards the domains. So it only is looking at the sequences. And then this allows us to look more into some novel proteins if we want to get some novel functions for these proteins and like annotate these proteins initially. So then here we see like we use an identity percent identity, how much is the sequence length, how much is the alignment length, what's the coverage, how many gaps were there. And this is the formula for the calculation of the discovery score. And then we have the knowledge score, which takes into account everything that is present in the discovery score, right? Identity, sequence, alignment length, number of gaps, coverage, everything. And then on top of it, it also has different parts of the formula that takes into account the domain alignment. So this means same type and order and number of motifs, same type and number of motifs, same type of motifs, or a no match, right? So when you have like same type, order, and number of motifs, that means these 2 proteins are very similar. And of course, the no match is like very different ones. And in certain cases, we have no predictions possible from some of the tools. So we have to handle that as well. So that's a part of the formula here as well. And this is very stringent because it considers all the possible information that we have on these protein pairs. Okay, so this is just an overview, like what we get, right? So we have 2 different transcripts, T1 and T2. So for them, first we get our identity, coverage, and gap information from the sequence alignment. From the domain alignment, we get different features. What's the type of the domains we have? What is the order of these domains? And what's the count of these domains? And then we have a calculation of our discovery and the knowledge score. And then from these, what we see is something very nice to see here is, so you see these pie charts. So KS represents knowledge score, DS represents discovery score. So only the protein pairs or the transcripts which have a score of more than 95. So a knowledge score of 0 to 100, right? So when it is more than 95, they were grouped together. So this big chunk represents different protein coding transcripts which come from the same gene, right? Like this is quite obvious, like we saw for the TP53 example. Out of those 25 transcripts, 13 were making the same protein. So this is quite okay that we have. And then we also have these ones, these green regions, so which represent a different family, like the example that I showed you with the histones or the ubiquitin conjugating enzymes. So these are still okay that we know about them. But then this small orangish part, pinkish orangish, so this is proteins that, the genes which come from different family, right? Like this is quite shocking for us as well when we saw that the transcripts made by genes which were not functionally like grouped together in any of the annotation databases, they still make the transcripts that make the same protein. Maybe these transcripts were not the major output of that gene, maybe a smaller transcript, but having a very similar function with the transcripts from another gene. So that was quite something which we found. And this is the same for all the cases we see. And then you can also see, like when we start lowering our score, so from 95 to 85 that we are going, right? So the proportion of these ones is increasing. So that means less similar transcripts are also getting grouped together, which we can see here as well now. So when we are making the protein pairs, right, at different scores, So we see these bars growing up, right? So a DS of 95, the bar length from 95 to 90, you see the change. From 90 to 85, you see the change. And the knowledge score is a bit more stringent, so that's why you see a comparatively a lower increase than the DS, right? So it's more stringent. Okay, just give me a second. I just got a notification that my battery is down. Just give me 1 second. I'm really sorry. I should have taken care of this. So yes, so this is what we have here, and then we see the group sizes is also increasing with the with the scores going low, and then this mean is also increasing a bit, right? So this is what we see here. Okay, so then coming to the calculation of the expression. So the thing is that the groups that we have created for each protein coding transcript, right? Like, so for each protein coding transcript, we create one group. So this is a dummy example that you see. So for transcript 1, we create a group T1, T2, T4. And then for transcript 2, we create a group T1 to T5. And then we see there's an overlap, right? Like these 3 transcripts are part of first one and also the second one. So then how do we calculate the expression for this? How can we really do that, right? So we really have to see that. So we use 2 different approaches to do that. So the first one is an equal distribution, right? Like when we have 2 transcripts in 2 groups, say this is expressed, it has 10 units of expression. So we put 5 to group 1 and 5 to the other one. So implying that both of these groups are equally important. So that's called equal distribution. And then another one is group size distribution, which means bigger groups have more important functions, right? Like when a group has more transcripts which have the same function, so that means this group is more important for the biological system, and that's why there are a lot of different transcripts having that type of function. So then based on that, we do a group size distribution, so the bigger groups get more chunk of the expression. Okay, so now coming to the assessment. So how we did that. So I'm sorry. So this, we have the RNA-seq dataset from 3D human hepatic cell models, and which was exposed to a PAP, also known as paracetamol. So I had 4 different set of samples. So we had an untreated control, we had a controlled DMSO, and then we have a therapeutic dose, and we have a toxic dose of a PAP. And these were the different time points we had for our data, for our samples. And there were a total of 54 samples, and we had a higher depth of the sequencing. So we had 41 million reads per sample on average, the largest being close to 50 something, and the lowest something around 35, 37 million reads. So that was quite good to do a transcript based analysis, because when you want to do a transcript based analysis, it's important that you have at least more than 30 million reads. So this is no written rule, but this is recommended. So you should have at least 30 million reads to do a transcript based analysis. Okay, so first we wanted to see that when we use our tool, right, the fuse, does it change how the samples are to each other, right? So when we use SFPG expression based analysis, how different our samples look. So when we do a normal from our data, the expression data we have, we do PCA plot, and then we do a PCA plot after applying fuse. You can't see the difference in 2 plots, right? So maybe you can see a bit of change in the 2 PCA components So 94.2 to 94.56 or something, 2.69 to 2.67. This is very minute, but this really means that the samples, how they look and feel with each other, this does not change that much. And then we also do a hierarchical clustering, and we also see that the same samples group with each other in both of these analyses. So this means that Fuse does not change the feel and look of the sample, and that's what we want. Okay, so then coming to the differentially expressed transcripts that we have between the tool and then the normal analysis that people do with the expression data, FPKM here. So what you see here is some of the Venn diagrams. So how you read these Venn diagrams. So when you have these two dots together, so this is between Fuse and FPKM. So this means that this is the overlap between the two sets. The individual dots represent each data set, right? This FPKM and this one is from Fuse. So whatever was left after the overlap, right? So what you see here is the overlap is 92, and this FPKM analysis we had 3,300 more differentially expressed transcripts, and with Fuse we have little less, not little less, like very less, 17 only. So this is what we see here. So the thing is that when applying Fuse, right, like you can see across all these comparisons. So this is a PAP therapeutic versus toxic, control untreated versus therapeutic, untreated versus toxic, and so on. So you can see in all these comparisons that the green bars are much, much smaller than the blue ones. So which means that always there were less differentially expressed transcripts with Fuse. Like you can also see that on the left-hand side, the Fuse bars is smaller than the FPKM bars, right? And then there is always an overlap, but still the numbers is pretty low. And this means that the transcript that it shows were changing, have changed, have been taken care of by the other transcripts in the group. So maybe one transcript is going down, the other one is up and still making the same protein. Okay, so even the ones that are grouped together, right, like these ones which are shown to be differentially expressed. So when we start looking at these, so what we see is even they change in direction. So maybe it was upregulated with the FPKM analysis, but when we use a Fuse analysis, so they were shown to be downregulated. So these orangish ones, these are a big part of these bars. And then these blue ones go from down initially with the FPKM and then go up with the Fuse analysis. So even if they're differentially expressed, but they're differentially expressed in a different direction. Okay, so this is about numbers, right? Like these numbers have changed, that these genes have changed, like how functionally relevant this is. So I will give you one example here. It's on the glycogen depletion. So we know about, like, when we take a PAP or paracetamol, then it has a, there is a glycogen depletion in the system. And this is one of the early biomarkers of a PAP-induced hepatotoxicity. So the glycogen has to decrease after taking a heavy dose or like a continuous dose of a PAP. And then through this example here for this particular transcript called PPP1R14B203, so we can see with the Fuse it shows it's going up, but with the FPKM, I'm sorry, with the FPKM you see this is going up, and with the Fuse it's going down. Right, also between DMSO therapeutic and DMSO toxic, you can see the same type of pattern. And this expression pattern, what we get with Fuse, aligns with our understanding of glycogen depletion in the system. And similarly, another one is GBE1, and this is responsible for the accumulation of glycogen in the liver. And with Fuse, we show this is also going down, which implies that the glycogen is getting is depleting in the liver. So these type of findings we could not have with the FPKM, but only when we apply a Fuse-based analysis. Okay, so this is another thing what most people do when they do a transcript-based analysis. So because they cannot take all the transcripts together, because every transcript is expressed at a different level. So what they do is they only take the longest protein coding transcript of that particular gene. And then here I want to show like how that is also not a good thing to do. So say in this case, when we talk about the gene expression, we see this is going down from untreated to the toxic dose for the longest protein coding one, it's also the same. But with the fuse, we show that the function of that the overall functional capacity of this particular transcript is going up. And similarly, in this one, we see the gene expression is going down, the longest protein coding is upregulated, and with the fuse, we also show it's upregulated. So I just want to show that like gene expression in the longest protein coding transcript do not align with each other, and at the same time, this does not align with the functional capacity that's given by fuse. Okay, so now finally coming to the conclusions of this talk. So instead of the chromosomal locus-based grouping, I was able to do a more functional-based grouping, where I take into account the amino acid sequences and also take into account the functional and the structural domains. And then we have two different scoring schemes with this tool, and then it's up to the user that how they want to analyze their data, so to fit their needs. And we also saw a lot of examples where transcripts change their direction of differential expression from going up to down or down to up, because the other transcripts in that group accommodated the expression of one transcript. And then this tool has a forward compatibility, which implies that any updates to the sequencing data for the amino acid and any updates to the domain information from the interpro, it can handle all of them. And finally, I would like to thank some people that were a part of this work. So this is Florian Kemont. So he was my supervisor. This is Jos Kenyan. So he was my co-promoter. And Yannick is the guy who did all the lab work. So generating all the wonderful libraries and did all the sequencing. And Masha was the postdoc who helped me with the bioinformatics work, guiding me at all different phases. And yeah, before I end, I just want to say this one thought which really stays with me. So when we learn something new, we do not go from wrong to right, rather we go from wrong to slightly less wrong, right? Like maybe this is just not the ultimate answer to the RNA-seq analysis, but at least we are moving away from the wrong. Okay, thank you. And I'll be happy to take any questions or suggestions.
Okay, thank you, Rajinder. Very well organized talk, I should say, because when you're starting from central dogma of molecular biology, and then you take your journey from transcriptomic data analysis, and finally to develop a very interesting tool, which is purely computational.
Yes.
It's really interesting to see your journey growing from like pure biotechnology student into a core bioinformatics here. Because I remember when you came here, you were having very less background in bioinformatics, but definitely now you are doing lots of work in this domain. So I'm really happy for you. And
yeah, I had my struggles in the initial phase of my master's. I remember that.
Yeah, because you need to be good in computations also when you want to survive in this area. But anyway, dear all, we welcome all your queries here. If you have any query, please submit in the chat box. Already we have, I think, 2-3 queries. So I will just read them. Rajinder, you can go through them. Rohit Sukla is asking.
Just 1 second, sir.
Yeah.
So the thing is, my laptop might die in a few minutes. So I will go on my phone and connect. Just give me 1 second. I forgot to carry my charger with me.
Okay, no problem.
If you give me 1 second, so I will be through my phone with the session,
Sure,
and
sure.
I can answer questions.
And dear participants, thank you for listening us. And I am just pasting you one link. This is actually the Google form link for feedback form. Please fill this form and you will get the certificate of participation. Thank you very much, those who are still here with us. And we are still continuing with this session. Please fill this form before leaving, okay? And if you have any queries, please ask now. We are here to solve them.
Sir, I just want to ask, though I
Okay.
joined a little late.
Yeah,
Hi,
no problem,
Rajendra.
ma'am.
Hi, sir. So I just want to say that he's doing a great work. I just want to know, dear, how would you like to say that it will help the journal masses if they would like to refer your work? How would you like to comment on that?
So what I would say is like they can use this tool in their RNA-seq analysis, and just because I could not do the analysis for all different data sets, so I would really appreciate if people can use this tool and share their outputs, and if they align with the functional knowledge about their tools or not. So that is the type of thing I would like to know. And this is the way I think people can be part of this, or uh, help grow this tool or something like that.
Okay, okay, Rajendra. Thank you for your answer. Thank you for your answer. Thank you, sir. It's a great
Okay,
session.
yeah, okay, ma'am, thank you. Okay, Rajinder, so here is one query that Rohit Shukla is asking. You have told that we require 30 million reads for transcriptomics analysis. So are this number like comes after pre-processing, or maybe remove lowly expressed genes or from raw NGS data?
So just give me a second.
Yeah.
Yeah, so yes, sir, I'm online now from the phone.
Okay.
Yeah. Can you repeat your question, sir?
Yeah, it's like you said that on an average, it is recommended that 30 million reads are required for doing transcriptomic analysis, as you mentioned in the talk also. So he's asking like, if this number comes after pre-processing of NGS raw data, or after removing all the raw reads or unrequired reads actually.
Yes, so this is recommended that when you do the initial filtration of your data, so this number should be after that, because it doesn't make sense to have the reads which are noise, right? Like the ones that you remove are usually the noise in the data. So if you remove that and you have less than say 20 million reads, it just doesn't align with the idea that the transcripts are well quantified. So it has to be after the filtration.
Okay, so it means the final number.
Yes, yes.
Okay, Rohit Supra, you want to ask something?
Yes, sir. Yes, sir. Thank you, sir.
Yes, go ahead.
So, sir, in the first step, like we did the trigonometric, we use the trigonometric, and we remove the lowly expressed genes, like that are not matched. But actually, my question is that, like you told that 30 million reads should be necessary for the RNA-seq data. So, like firstly, we will align, and then when we got the count file, and after that, when we will use HR or some DE-sec2, and then that step when we will remove that, okay, with 0.5 cpm we will remove all the reads. So is that criteria that in that number 30 million reads are come, or in the previous experiment, like when we are doing the fast QC and other analysis?
Yeah, in the initial stage,
In the
not after
initial.
you do. Yes, yes.
Okay, okay. Thank you, sir. Thank you, sir.
My pleasure.
Okay, sir. One more
Hello,
question is.
Rajendra.
Okay, sir. One more question is there, that have you used any machine learning tool in this, or any machine learning method in your tool, FUSE? FUSE?
For this tool, the machine learning part that I used is for the domain predictions, the functional and the structural domain predictions. So there are some tools which depend on machine learning algorithms, and these are a part of Interpro. So this is the side for which I only use the machine learning.
Okay, but you have not used, like you have not evaluated your tool in accuracy type. I'm just asking, sir. Actually, I am working on same type of project, so that's why I am just taking the idea.
Yeah, yeah, that's totally understandable. No, for the evaluation, towards the final evaluation of the tool, so no machine learning was used. It was more towards like what functional characteristics we understand for the APAP. With that respect, we try to evaluate this tool. But of course, that's a good thing that we should have done it through machine learning thing. Maybe if I develop the next version, I will have that thing in my tool.
Okay, thank you, sir. Best of luck, sir, for next
Thank
one.
you very much.
Okay,
And
okay,
good luck for
Rajinder,
your
I
work.
think Neha Goyal is raising hand. Neha Goyal, if you want to ask anything, please ask.
Uh, yeah, hi Rajendra. Uh, Actually, I have a query not regarding your software, the Fuse, which I'm already tried in my work. Sorry, you Actually, I'm already working in the prostate cancer. So, and I have a query regarding because I got the 8,000 significant RNA-seq data and I'm stuck with the heat map. When I'm making it, it is really a bullshit map, I should say. And it's like a mess. And I'm trying with the criteria of p-value 0.05. Even I'm not, and when I tried with the K clustering in that, K-mean clustering, that time I'm getting the genes, the larger number of genes in one cluster. Now I'm stuck somewhere else. Okay, what should I do to make my heat map in the visualized format? So by not by the k-mean clustering, I'm not satisfied with this. Is there any way to do that?
Yeah, so one suggestion that I would give is like when you have such large number of genes, right? So what some people do is like what you have already mentioned is like they start increasing the adjusted p-value, the q values for the filtration. But if you don't want to do that, so one way is to start grouping your genes based on their functional annotation and start plotting the heat maps based on the functional annotation of genes. In that way, you would always also have a better understanding of some functions, right? So say for a functional annotation of say transport genes or something, so you have 1000 genes there rather than 8,000, so your heat maps will look up much better there, and then you can evaluate like how the transport mechanisms are impacted, or you have some other functional annotation of the genes and you can start grouping them and then make heat maps for them. I think that will be an
So
initial suggestion.
for that way, I have to take, I have to select some specific pathway for that. I think for the functional annotation, as I already did for the, actually I got 27,000 genes, and after that filtering with the p-value and the log fold change value, I got 8,700 genes. And the functional annotation I have done for the gene ontology and the KEGG analysis pathway for that. But the problem is that again, when I'm going, I have to select a specific pathway to make the heat map. I think that will be the better way.
Yeah, so one thing is to do pathways, but I would say like most of the times, if you look at different pathway databases, you would see a huge, mismatch between the same pathways throughout these databases. So you really have to make a selection what database you choose for your analysis. What I would do is like you can get gene-based annotations, right?
Yeah.
From so and then based on these annotations, gene ontology terms or something, right, at different levels, gene ontology terms that you can use, and then you start
Yeah.
grouping based on your ontology terms. So you might end up with, you have 8,700 genes, right? Like, and then this might end
Yeah.
up in, say, 10 different groups or like 15 different groups, and then create 15 different heat maps for these, so that you can, at one point, you can start talking about one annotation, one ontology term. So maybe that will help you. And then if you have some ontology terms which are connected with each other, say for instance transportation and translation, right, and
Yeah.
then you can see like two heat maps and make a comparison. That way you will have some path forward, because if you want to make a plot of more than 3,000 4,000 genes, it will just not make sense. It will just look like some color patches here and there. But
Hey,
if you
yeah, that's a really, so I don't have words like for this. It's like that when I was doing a hierarchical clustering on that, even the clusters are overlapping to each other. So it's like just aligning, just a black paint. I did it. It's more that it's not a cluster. Even I cannot see the clusters in that.
Yeah, like my initial suggestion would be to do grouping on the ontology terms. Yeah, but
So it will
even
be
if you
three groups
want,
together. It will be the three groups: the molecular process, molecular functions, biological process, and the cellular components. And these three groups are interconnected with each other, like as the mitochondrial pathways. Some cellular components participates in the molecular functions also. So is it possible to remove those genes, or I have to take those genes in both groups?
I won't be able to like answer that thing like right now. So maybe if you want, you can send me an email and we can have this
Yeah,
conversation afterwards, and then we
yeah, we
can
can do
see like
after
what can
this
be done
also.
about it. Yeah.
Yeah, sure.
Yeah.
And okay, sure.
Okay.
Thank you.
A
Okay,
pleasure.
Rajendra, there is one more query from Santosh Patra from Odisha. He's asking about like how metabolic disorder may be traced out at molecular level of study. It's like
Okay.
a different question, but if you want to
Yeah,
answer.
so metabolic disorder. So I think for that, you need a different set of data to start off with. So you need to have a metabolomics data. And then on top of it, now there are a lot of different proteomics approaches which are quite good. And then you can have a multiomics data set from a proteomics and the metabolomics data. And through that, it should be you should be able to get at certain conclusions about these type of disorders.
Okay. And there is one more query from Ajay. He's asking, like, after doing the RNA-seq analysis, what type of results we perform? And additionally, he want to ask how we can integrate machine learning techniques also.
I did a lot of different things with the RNA-seq data. So there is another paper out that I did for different liver cell models. So in that you can see I did some very basic analysis, what people prefer to do, like a pathway-based analysis, like DEGs, differentially expressed genes, differentially expressed transcripts, and then some ontology-based term analysis. And then also I did some cellnet analysis. So there's a whole lot of things which I think it will take a very long time if I start discussing everything about it. So, but there's a paper out there. So it's on the liver cell comparisons. So if you start looking at that, you will find some good starting points, I would say, what things you can do. And then with respect to the machine learning, so I did another work with RNA-seq data where I explored probable or like novel new biomarkers for liver cancers. And that's a part of a machine learning work which I did, and where we found out protein coding and non-coding transcripts, their expression patterns can be used as novel biomarkers for liver cancer. So that was also one study which we did with the RNA-seq data and more towards the machine learning side.
Okay, I hope Ajay got his answer. So I think that's all for today, Rajinder. We have all the queries for now, and I would like to just give a message to all the participants that please fill the feedback form before leaving. And I would like to thank each and everyone for listening us patiently. And first of all, I would like to thank Rajendra especially for sparing his time with us and then enlightening us with his knowledge that he gained in last like 1 decade after doing lots of hard work. And thank you very much. Thank you one and all present here. And we are going to organize another webinar next month. And I hope you all will enjoy that again, like today. Thank you very much. Thank you all.
Yes, so before you go and everyone leaves, so I would like to thank you again, sir. First of all, for this invitation, and second for all the support from all these years, and then always answering my queries about all the scientific and personal issues. You have been a great support throughout, sir. So thank you very much.
Okay, you are always welcome.
Yes, sir. And if anyone wants to ask or get in contact with me for anything, so they can send me a message on email address, or maybe, I don't know, maybe Tiratha sir can help you find my email address, or you can find me on LinkedIn.
Sure. I will share with you. And Santosh ji, yeah, date is not yet defined, but soon we will communicate with you all, okay? Because you are all registered with us, so we will send you the details of the next webinar. Thank you very much. Thank you, everybody. And thank you, Rajendra. See you soon. Okay, bye then.
Yes, sir. Bye-bye. Have a nice day, everyone. Bye.
Yeah, thank you.